Assalamualaikum to fellow lecturers and fellow classmates. We are from Group 4 going to present about the purple corn. So the team members are Muhammad Daniel, Muhammad Nul Ashraf, Muhammad Aslan, Muhammad Ilfan and Muhammad Afandi. The introduction. Purple corn, with its unique anthocyanin pigments, has potential economic value in food and pharmaceutical industries. However, breeding for improved traits is still in its early stage. This presentation will explore the biology of purple corn, the breeding approach, and the justification for the chosen approach. So, by understanding the plant's biology and using innovative techniques, new varieties with improved traits can be created, benefiting farmers and consumers seeking more healthier and natural products.
Economic important for peppercorn. Uses for food industry, export potential, animal food, and tourism. For value, high demand for having antioxidant. Volume, the Department of Agriculture record only 17.7 hectare of peppercorn planted in Malaysia and cultivated by 8 farmer alone in 2020. It is only for self-sufficiency and not for commercialization. The record from Department of Agriculture Malaysia, the production of peppercorn.
Trait of interest to be improved. Pest resistant. Developing purple corn varieties that are resist to common pests such as corn borer using Bacillus thuringiensis, known as BT. Corn borer belong to the family Noctuidae. These pests tunnel into corn stalk and feed on corn ears, leading to reduced plant vigor, stalk breakage, causing damage to kernel and increased vulnerability to other disease. With this improved trait, it can help to minimize yield losses and reduce the need for chemical intervention. Bt. Bt is a naturally occurring soil bacterium that produces insecticidal protein. These proteins are toxic and usually used as pesticide to control corn borer. Bt corn. Bt corn is created by inserting selected exotic DNA into corn plant genome to make the corn resistant against corn borer. The genetic modification involves isolating the specific genes that are responsible for producing the insecticidal protein in Bt and inserting them into corn DNA using gene gun. Successful process will create a corn plant that produce Bt protein naturally and provide resistance against corn borer.
Reproductive system. Peppercorn is a monoecious plant, also called hermaphrodite. The plant have both male and female flowers, which are located on separate parts of the plant. The male flowers consist of tassel and elongated spiky structure that is found on top of the plant that produce pollen, while female flowers develop into ears of corn, or also known as cob, with thread-like silks that connected to the ovary. Pollination can happen when wind or insect transferring the pollen from male to female flowers. The pollen will travel from silk to the ovary, thus developing the kernel. Propagation method. Seed propagation. It is similar to regular corn where it is propagated through seed. High quality seeds should be chosen to make sure the plants are free from disease and also will have high rates of germination. The methods of propagation is the seed of the purple corn is directly planted in the ground about fingertip depth and space between plants are about 24 to 30 inches. The plant is expected to be matured in 60 to 90 days. It is also important to handle pest and disease management as the plant is one of pest favorites such as stem borer.
Hi, my name is Muhammad Irfan Mizani Sham. Now I will continue about the appropriate breeding approach. The appropriate breeding approach that we will use is genetic engineering techniques. For the first one, we have identified the target pest. The target pest is corn borer. The BT toxin specifically targets and affect the larvae, or we call the caterpillars, of corn borer when they feed on the genetically modified corn plants expressing the toxin. And then select the plant species and cultivar. Select the purple maize variety that we want to genetically modify to express the Bt toxin. Ensure that the chosen variety is amenable to genetic transformation techniques. And then isolate the Bt gene. Extract Bt gene from Bacillus thuringiensis to produce effective toxin against identified pests. And create a plant transformation vector. Introduce plant transformation vector to maize cells using agrobacterium mediated transformation transforming cultures. Create a plant transformation vector, a circular DNA piece carrying the Bt gene and regulatory sequence for proper expression, and then transform the plant cells. Introduce plant transformation vector using agrobacterium mediated or biolistic techniques, inserting Bt gene into plant genome, and then screen and confirm Bt expression. Verify gene expression in transformed plants using molecular techniques like PCR and protein expression assays. And the last one, evaluate the effectiveness. Assess Bt toxins effectiveness through laboratory and field trials, observing genetically modified plants resistance and evaluating pest damage reduction.
My name is Muhammad Aslan. I will present about justification of the chosen breeding approach. To achieve the goal of strengthening pest resistance in corn plants, genetic engineering technique are the appropriate way. Genetic engineering technique aim to insert Bt toxin into the corn stalk system where Bt toxin is a bioinsecticide to fight pests while also increasing the ability of pest resistance in corn plants. To support this statement, studies report that BT corn has saved farmers millions of dollars each year because of the reduction in pesticide need to deal with corn borer infestation. According to SR Ekov and SC Young, 2003, 25% of the corn area plant in the United States in 2000 was genetic modification, also known as GM. In addition, according to a recent study, cultivating BT corn enhanced agriculture revenues by more than $8 billion in the first 13 years from 1996 to 2008, following commercialization. Thank you.